When we learn about cells, we often talk about what's inside of them, but how much do you know about their surface? Today, I'll be talking about the structure of the cell membrane, also referred to as a plasma membrane. A cell membrane is 10 nanometers thick and is comprised of two layers of phospholipids, called a phospholipid bilayer. Each phospholipid is made up of a polar hydrophilic, or water-loving head, and two nonpolar hydrophobic, water-hating tails. The head is made of phosphorus, and the tails are made of two fatty acids connected by glycerol. In a phospholipid bilayer, the polarity of the phospholipid is important. The head of the phospholipid is hydrophilic, so it will always be on the outside, whereas the hydrophobic tails will face inwards. Membrane proteins are another main feature of plasma membranes. Peripheral proteins are located on the inner or outer surface of the phospholipid bilayer. Integral proteins can either be embedded in some or all of the bilayer. Channel proteins can span across the lipid bilayer and form a hydrophobic interior in the membrane to allow water-soluble molecules like oxygen to pass by facilitated diffusion. Carbohydrate chains can be found on the extracellular side of the cell membrane and can bind to lipids to form glycolipids, or to proteins to form glycoproteins. They are important in membrane stability, cell signaling, and acting as receptors for hormones and neurotransmitters. The last part of the plasma membrane is cholesterol. It is a type of lipid embedded in the membrane which helps regulate the fluidity of the membrane. Membrane fluidity is very important as it makes endocytosis and exocytosis possible. This is the fluid mosaic model, discovered by Singer and Nicholson in 1972. Hence, it is also dubbed the singer-Nicholson model. Prior to their discovery was the Davson-Danielli model, which suggested that the phospholipid bilayer was sandwiched between proteins on either side. However, this theory was falsified by three main pieces of evidence. First, when membrane proteins were tagged with fluorescent markers, they were discovered to be mobile, suggesting that the membrane proteins could move. Membrane proteins were also discovered to be insoluble, indicating that the surface was hydrophobic. Finally, when the membrane was split open with a method called freeze fracturing, scientists found a rough surface, which were believed to be proteins embedded through the membrane. To review, this video talked about the main components of a plasma membrane, including phospholipids, membrane proteins, carbohydrates, and cholesterol, and also provided evidence falsifying the Davson-Danielli model. I hope you found this video helpful, and I'll be making more videos soon on other biology and chemistry topics. Let me know if there are any topics in particular you'd like me to cover in the comments below.